thank you very much for the invitation. I'll talk about that in a second. I just wanted everybody to know that if you're interested in these slides, and I maybe go through them too quickly, or you want to focus in on some of the errors that are in my slides, they're available online already. If you search for OSF Titus Talks, you can find the one for today. So please, and also please feel free to tweet about my talk if you want. I think that's an acceptable use of the network. And my handle is Titus Brown. Right, so first of all, thank you very much for the invitation. This is my first trip to Russia, and I've been enjoying St. Petersburg. I was in downtown. I don't know what the right term is, over by Nevsky Prospect for 2 days, and I got to see the new Hermitage and some other amazing buildings and city parts. So it's been wonderful. And then I've also been I'm also really enthusiastic about this meeting. It's very rare that I get to talk about both bioinformatics and biology to an audience that cares about both of them. So I'm really looking forward to the talks today and tomorrow. And I'd love to chat more about what everybody here is doing. So I want to start just by thanking my collaborators and of course the funders, and Microsoft. So the work that I'm going to talk about today is done largely with a graduate student in my lab named Taylor Reiter, who is from the Food Sciences Department, and then also 3 collaborators from North Carolina State University, Blair Sullivan, Felix Reidel, and Mike O'Brien. Mike is now at Google, and Felix is at Birkbeck University in the UK. And then Dominic Moritz at UW Seattle, who's a graduate student that worked with us. And I show some results from Harriet Alexander, who's a former postdoc who's now a professor at Woods Hole Oceanographic Institute. And I'll annotate the slides with the appropriate names. Most of the work today was funded by Gordon and Betty Moore Foundation, but of course I also have been generously supported by 2 of these 3 funding agencies. Okay. So I'm going to tell you a story. The story's not done. I don't know that stories in research are ever completely done. And if they are, then people generally don't talk about them anymore. But this story is particularly unfinished. And I actually have to say that I have a lot of fear and uncertainty and doubt around the relevance of what I'm doing here, because I actually don't know if it matters. So I'll just start my exciting talk that way. I think I know that it matters computationally, but I'm not sure it matters biologically. So, and I think there's some other talks here today that will maybe help me figure out whether it matters biologically. But it is pretty cool computationally, so I have that going for me. I wasn't sure how many people in the room would be familiar with shotgun metagenome sequencing. Looking at the list of talks, it seems like there's a lot of expertise in sequence analysis in general. So I'm going to have one slide on shotgun metagenome sequencing, which is this slide, which basically says shotgun metagenome sequencing here is what I'm talking about is taking complex microbial systems and sequencing them directly without any culture or other selection process. And the idea is that you take DNA from some environment, you fragment it, you do something horrible to it molecularly, and then you sequence it, and you get out a bunch of data that goes into the computer. And usually at this point, you leave it up to the bioinformaticians to figure out what to do with the data. So the key points here are just that we're going to be looking at mixtures of microbes. So what's in the sequencing data is not a single genome, but it's many genomes, some of them closely related to each other. Okay, so the story starts with when I started as an assistant professor at Michigan State University back in 2008. And I arrived at Michigan State University and I had very little experience with metagenomes at the time. I'd done a little bit of work with Victoria Orphan at Caltech as a way to escape doing my primary thesis work. I did sequence analysis on the side because my primary work in developmental biology involved a lot of experiments, and experiments can be very boring. So instead of doing the experiments, I went and did sequence analysis for other researchers. And so one of these was a 454. You might remember the technology. 454 metagenome sequencing data set that we published, I think, in 2008 or 2006. But that was my only experience with metagenomics at the time. I arrived at Michigan State, and immediately I started working with Jim Teege, who's a soil microbiologist. And Jim is one of these many biologists that had an amazing capacity to generate many extremely large sequencing data sets. These days, that's kind of normal. Back then, it was a little less usual. This was 2010 or so. And basically, I think for those of you who were doing active research in 2010 and 2012, Illumina GA2 and then Illumina HiSeq had come out. And if you were doing anything that wasn't reference-based, anything that wasn't sort of biomedical, you knew that the tools basically just didn't work. You would get all this sequencing data, you'd feed them into the computer would melt down, you'd adjust some parameters, you'd try it again, the computer would melt down, and you'd sort of repeat a couple times. And so Jim, together with Susanna Tringe and Janet Jansen, had generated this extremely large, for the time, soil metagenome data set from 8 different agricultural and prairie soil samples. And so in 2014, for what I think was my second paper, we managed to successfully assemble these data sets. Successfully is an interesting word to use with assembly. We managed to run the assembler and we got some output and we managed to publish it. So I'll call that success. And basically, we outlined a workflow that nobody should use anymore for eliminating some k-mers and doing some other clever stuff with the sequencing data to sort of get it to the point where the computers that we had access to would actually complete the assembly. So, and one of our questions for this work was, is there a lot of strain diversity present in the data set? Because we knew that soil should be tremendously complex. We knew that there should be a lot of closely related bacteria in there. And so we asked, one of the questions that we asked ourselves, but we didn't end up really putting in the paper, was, do we see many different closely related bacteria in soil or not? And as you can tell from the high quality graphics that I have here, I've lost the primary data. This was another institution. This was many papers ago. This is a graph I generated for myself in 2012, and I just couldn't find it anymore. But what I remember is that we to look at strain variation, strain diversity, we looked at the mismapping between assembled contigs and mapped reads in soils. That is, when you took the assemblies and you mapped the reads that went into the assembly back to the assembly, did you see a lot of differences or not? And in general, we found that no, we didn't see many differences, which is to say, I remember a number around 1 or 2%, something in line more or less with the GA2 sequencing error rate, that when we mapped the reads back to the assembly, in general, the reads didn't have any mismatches, serious mismatches with the assembled contigs. And we'll come back to this graph. There's a little feature here that I conveniently ignored at the time. And this talk is basically about this little feature. So the conclusion that we reached from this paper in 2014 is that what really mattered for metagenomics was sequencing coverage. If you could go and you could get some DNA and you could sequence the heck out of it, do an awful lot of sequencing, then you could get an assembly from it. And this was borne out by a certain amount of evidence. One in particular was when we looked at the mock community, you saw that maybe I point over here. You see that low coverage stuff didn't get assembled, but high coverage stuff did get assembled. And we saw these curves here from our data set where we saw that we simply didn't see low coverage stuff based on medium k-mer coverage of the contigs, but we did see a lot of high coverage stuff. This was using, for those of you that, again, were doing this kind of research in 2012, I think we ended up using the Velvet assembler. After a bunch of pre-processing to even out the coverage, I just saw our EBI representative nearly faint at the thought that somebody used Velvet seriously, but back then it was a really good assembler. Plus we did a lot of good pre-processing, so it's okay. Yeah, so we sort of said, well, look, what we need to do is shift this curve to the right by doing a lot more sequencing. That's essentially the solution to metagenomics in our view. And the paper was, more or less written in 2013 and so on. Okay. So I remember going to a conference in 2014 and telling biologists, look, like Metagenome assembly works. It's mostly a matter of coverage. Jill Banfield said something similar at the JGI, like if you just sequence more, then it's all going to work out. And I said things like assembly mostly just works. Okay. This is probably the right audience to laugh at this, but you know, it was a reasonably tendentious statement at the time because a lot of people at the time were still analyzing single reads, individual reads. You know, 80, 70, 80 base pair Illumina reads to get at the content of their metagenomes. And since then, we've gotten better assemblers, we've gotten better workflows, we've gotten better sequencing data, but and we sort of have some level of cross-validation. So we think that assembly does actually kind of work now. Maybe. We'll talk about that a little bit more. So I made these statements at a conference, and nobody laughed, and all the biologists nodded and said, great, that's wonderful. We just need to spend more money on sequencing, or wait for Illumina to double their sequencing output, or wait for Nanopore, or whatever else was going to happen. Okay, so then I got a postdoc in my lab, Shireen Awad, and I said, hey, like we have some clever tools for processing sequencing data. Why don't we look at how they respond to assembly? And the story ended up being really much more about assembly than about our tools. And we took this mock metagenome published in 2013 from Mircea Podar's lab, which had 64 species of bacteria and archaea. This was a mock metagenome. These bacteria and archaea were grown in the lab individually in clonal isolates, and then combined and DNA was extracted. DNA was extracted individually, and then they were mixed in a defined ratio, and this was meant to be a benchmarking data set. So they did 16S sequencing with 454 and Illumina, and they did shotgun metagenome sequencing with 454 and Illumina as well. And the one thing they didn't examine in this paper was how well assembly worked. They had the reference genomes, they did mapping, they did coverage analysis and everything. And it's a really great paper to read if you're interested in just how bad our bioinformatics tools are. Because what they show is that there's sort of like some good correlations between what the tools produce and what was in the data, but the correlations aren't that great. I think they've improved since then. But so our main result from this, so we took this data and we passed it through an assembler and through 3 assemblers actually. We assembled it with MegaHit, IDBA, and Spades, and basically we had ground truth, more or less. We had the source genomes that went into this metagenome community. They'd been fully sequenced. There's some interesting stories there that I can tell you later, including some overconfident statements in the Spades paper that I'd be happy to talk to Anton and others about. But basically, for this mock metagenome, we could calculate the NGA50. That's the aligned NG50 against the known truth, right? We could assemble it, we could take the contigs, we could map the contigs, we could say, how well did these assemblers do? And we noticed a real dichotomy. Out of the 64 metagenomes, there were 64 genomes in this metagenome, there were about 15 that had NGA50s of less than 10 kb. That's pretty fragmented for a community that is, at the time, for a community that didn't have much complexity and had been sequenced quite deeply. You would have expected something bigger, and in fact, most of the genomes were down here with NGA50s of between 10 and 100 kb. And so basically we noticed this big divide. Some genomes were recovered really well, and others were recovered in many small fragments. And it didn't really seem to matter what assembler you used. Some of them just failed miserably. Others sort of succeeded somewhat. Spades, you'll be happy to know, was often the biggest. But by and large, we saw this pattern. And when we looked further at the pattern, what we saw was that when you did a Jacquard dissimilarity matrix, that the genomes that were assembled poorly by the metagenome assembler were the ones that had one or more companions in the metagenome. That is to say, the genomes were within, say, 1 to 2% Jacquard similarity of each other. And all that really means is that these were not distinct species, these were strain variants of the species. And this was more or less a perfect correlation. Anything that had a star was something that had a very close relative in the metagenome. So anywhere where you saw sort of more than a single perfect genome here, these blocks are things that assembled poorly. Okay. So basically, what we reached, this is a paper, I think, did I put the reference in? It's up on bioRxiv. Our conclusion from this is that strain variation in metagenome assembly is actually treated like a high error rate. When you have a lot of k-mers that mismatch in a particular region, or contigs aren't formed, and when you actually look at the assembly graphs that are sort of the pre-processed assembly graphs in the metagenome assemblers that megahit, for example, outputs, you actually see that these k-mers are just completely missing from the genome, from the graph. And that seems to be because during the error correction phase of metagenome assembly, the metagenome assembler says, this looks complicated, it's probably wrong, we're going to ignore it from now on. So the assemblers don't say, here be dragons, the assemblers say, I can't do anything with this, I'm not going to output anything. Or to put it a little bit more succinctly, strain variation seems to break assembly. This is well known from genome assemblers, where if you have a lot of heterozygosity in a diploid genome, those regions also tend not to be output, or used to not be output as well. Okay, so now if you go back and you look at this graph, maybe this graph of where mismatches occur between reads and contigs looks a little bit different. Basically, this rise here suggests now to me that, and if I'd thought about it more then, might also have suggested it to me then, that actually what's happening is as soon as, in a metagenome, as soon as the reads start to get, come from regions that have a lot of heterozygosity or polymorphism, the assembler stops and breaks the contig and throws away everything else. I count this as one of the top 5 mistakes in or missed chances in my career so far, because if we'd thought a little bit more about it rather than just trying to get the paper out, we might have had something more interesting to say than, hey, we managed to run the assembler. But, that's life. So, so the rise in variation towards end of contigs really suggests now, when I look back at my memory of this graph, that strain variation might be breaking the contigs. Okay, wow, that didn't, okay, well, in lower resolution, that looks a lot better. I'll just say that when we go into metagenomes and metagenome assembled genomes in particular now, we actually see that nucleotide assembly performs very poorly from a k-mer containment perspective. I'll come back to this later, but basically we see that sometimes as low as 5 to 20% of the k-mers that are in the reads underlying a metagenome assembled genome or a genome bin don't make it into the, 80% don't make it into the genome bin. And I'll talk more about this later. But basically, we see a lot of variability in which parts of metagenomes assemble well and which don't, and maybe that's related to strain variation. Okay, so the story so far. So a long time ago, I ignored some signs that assembly might break on strain variation. When we did some benchmarking, we saw that closely related strain variants interfere with each other's assembly. The JGI reached the same conclusion from some synthetic data sets. So basically, it looks like strain variation breaks assembly. So this all sounds well and good and is a great conclusion to reach and makes a lot of sense, and I could probably convince you standing at the front of a room, but how do we look at strain variation without doing assembly? Right? If we're looking at environmental data sets with no reference genomes, it turns out to be extraordinarily hard to look at completely unknown sequencing data sets with tens to hundreds of millions of reads without running the assembler first. And so it actually took us about a year to figure out what to do with this, and I'll get to that in a bit. Okay, so the question is, how do we look at strain variation without doing assembly? And our answer is we inspect the assembly graph. And so this is, for example, I just wanted to put up a picture of a graph because I'm going to be talking about graphs. And so this is actually a picture of a compact de Bruijn graph of simulated E. coli read data. As you can see, it's hard to lay them out in 2 dimensions, and they look really complicated. And they get really big when you're talking about metagenome graphs. So basically, I spent 3 years working with some graph theorists to figure out how to index and query sort of arbitrarily large compact de Bruijn graphs. And this was the collaboration with Blair Sullivan, who was at NC State and is now moving to University of Utah. She's a theoretical computer scientist who works on sort of the tension between algorithmic complexity and practical implementations of those algorithms. And I have to say that part of the reason it took 3 years is that it's actually, I know this is being recorded, I think she would probably agree with me, it's very hard for bioinformaticians to talk to serious graph theorists. I would argue it's very hard for anybody to talk to serious graph theorists. She would probably have the same comment about bioinformaticians, but in the end it turned out to be a very productive collaboration, and I think we can now talk to each other more easily. And this project resulted in a bunch of code called that we call spacecraft cats. If you want to know why it's called spacecraft cats, there was a German postdoc, Felix Riedel, involved, and I said I would like to figure out how to cut sparse graph cuts, and he free associated, he likes cats and graphs, so he free associated to space graph cats. You got to name them something. It might as well be something memorable, space graph cats. So what we did was we worked out a procedure to go from reads, and here we have sort of an idealized version of reads, or contigs, or whatever, what have you, full genomes, to a compact to brain graph. And here we actually just took advantage of the BCalm, the wonderful BCalm CDBG creator. And then we go from these compact to brain graphs, and we calculate what a set of dominating nodes. And what dominating nodes are is a fairly straightforward graph theoretic notion that you can pick a subset of these nodes such that every other node within the graph is within some given distance d of that subset. So here, if you look, every gray node in this graph is within a distance 1, is immediately adjacent to a purple, reddish node. And so there's a dominating set distance of 1, a dominating radius of 1. And the idea is that these pink nodes can serve as sort of a proxy for the graph structure. They're a much reduced or compact or compressed representation of the underlying graph. And you can go from a graph that contains 10 or 20 or 30 million nodes to a dominating set that contains in the neighborhood of 100,000 to 500,000 nodes. So you get a really dramatic decrease in representation size. And so this turns out to be an NP-hard problem. I'll talk a little bit about that in a second. And we can do it to extremely large graphs, sort of arbitrarily large graphs, human genome-sized graphs in sort of seconds to minutes at this point. Then we take these dominating nodes and we use them to define neighborhoods. And the idea behind these neighborhoods is that you take some sort of query, say query 1 or query 2, you find all of the dominating nodes that cover that query, and then you go back to the original graph and you extract all of the original parts of the graph that now belong to that expanded neighborhood. Right? So it's sort of like, I used to have all these pictures when I was first talking about this, of like sort of landing on a house in a neighborhood and then saying anything that's within sort of 3 blocks of that house now is part of that house's neighborhood, and so you sort of go from single points to expanded points. And then we ask for a given contig or genome, these are these queries, what reads belong to these neighborhoods that weren't assembled? Or actually, what are all the reads in the neighborhood? So the whole point of this 3 years of work that we did was to go from an assembled contig or an assembled genome or a k-mer, doesn't really matter, back to all of the reads that maybe should have been assembled into that portion of the graph. And some of them were and some of them weren't. Okay, so first of all, I want to impress you all. This is not my work, this is my collaborators' work. Basically, finding a minimum R dominating set is NP hard. And in general graphs, you can't find good approximations. Because de Bruijn graphs, compact de Bruijn graphs have a special type of sparsity called bounded expansion, Blair's group was able to implement a linear running time approach for calculating a dominating set for these graphs. So literally we have a pure Python implementation that runs on millions of nodes where the dominant cost is loading the data, loading the graph from the disk, and everything else runs faster. So somewhat to my chagrin, we have many thousands of lines of code in this data set, and by far the fastest is the hardest part, which is the dominating graph calculation. Everything else is like walking through reads and sorting things and extracting reads and so on and so forth. So this is actually something that I suspect people who are really interested in large graphs, including assembly graphs, but also protein-protein interaction graphs and so on, might find interesting. Okay, so we have a whole workflow, and I mostly put this up to show you that we have a whole workflow and it's all implemented and it's open source. But at the end of the day, what we end up with is this. What we have is we have a set of reads from, say, a metagenome, and we have a query sequence that we want to go into those reads with, and we can use that query sequence to retrieve all reads within some graph distance R of the query. So it's as simple or as complex as that. It's simple in concept, and it's actually a lot of code underneath, but it seems to work. So, right. So this includes all of the unassembled reads. Not all of the unassembled reads in the metagenome, just all of the unassembled reads that are close to some known contig or genome. Okay. So then we decided we had to look at some real data. We did a lot of work with synthetic data sets, and that all worked. And we said, okay, well, what do real metagenomes look like? And so we took a data set from the Banfield lab that the Banfield lab had published in 2016. This is an oil well data set. And they basically generated about 20 bin genomes from this SB1 sample, and they used a fairly standard metagenome assembled genome approach where they took the data, they took the shotgun data, they assembled, then they binned the contigs by sequence characteristics, evaluated the completeness, and analyzed their metabolic potential, right? So they're going from a metagenome to about 20-odd curated high completeness metagenome assembled genome bins, MAGs. And we were often asked why we picked, Jill Banfield asked me, like, why did you pick this data set? Of all of the interesting data sets that my lab works on, I would not have picked this one. Why did you pick it? And the answer is it was small. It's only like a couple 100 million reads all told, and we could do the whole thing without sort of spending weeks and months running assemblers and consuming all the memory available to us. So this is our first metagenome that we've analyzed. So we actually don't know what this does on more complex metagenomes. On larger, more complex metagenomes yet. Okay, so the thing that I want to highlight in red is our entire genome binning workflow these days depends on assembly, right? So when you go into a metagenome and you extract genome bins, what you're doing is running an assembler and then working with the contigs that come out of that assembler. Okay, so the first thing that we noticed is that neighborhoods in real metagenomes are big. So when we go in and we look at the number of k-mers that are in the query, and we compare that to the size of the neighborhood that's extracted from that query, we see that essentially half of the genome bin, half or more of the genome bins have in their neighborhood 50% or more additional k-mers beyond that bin genome. Sorry, I'll try and stay over here. And so basically, when you go in with a metagenome, you go into a metagenome with a genome and you pull out all of the reads that sort of are in the proximity of your query, you get a lot more out than just the genome. This is not true for mock metagenomes. For mock metagenomes, you go in, that's right, yeah, you go in with the genome and you pull out all the reads, and most of the reads basically map directly to the genome. For real metagenomes, there's a lot of stuff that doesn't make it into the genome bin. So neighborhoods in real metagenomes are quite large. And this is actually where this table comes from, where we see that after soft trimming the reads for erroneous k-mers, we get at best around 50 to 60% assembly of the k-mers into the genome bin, and at worst we get something in the 5% range. This is after we extract the assembly, we extract the reads, and then we assemble them with megahit, and we get sort of a range of assembly parameters, assembly results. So now we faced a problem. Somewhere between approximately 50 to 80% of the reads that we got out of the neighborhoods weren't assembling. It's probably why they didn't make it into the genome bins in the first place. That's why the neighborhoods are so big. And some of them are certainly errors, right? I would be hard pressed to say that even after our soft trimming, probably a solid 20 to 40% of the k-mers in a given neighborhood are just erroneous sequencing errors, especially for some of these high coverage metagenomes that are in this data set, high coverage genomes that are in this data set. But when you get down to 20, it's the variation here. It's the fact that some of them are 50% and some of them are 20% that sort of got us wondering what was going on. But now we face the problem. What do you do with stuff that doesn't assemble? Turns out to be quite hard, especially when you have many millions of reads. And we had gone into this with the suspicion that strain variation was the problem. We knew it probably wasn't coverage, because this was a very high coverage metagenome of a fairly biased community. So we expected that we knew that some of these had coverages of 200 or 300x in the source genome. So we tried a lot of different things, and what we came with at the end was maybe if we do assembly in amino acid space, these things will assemble better, right? And the logic underneath is hopefully clear if you work with assembly, which is just that many polymorphisms don't affect the coding sequence much or at all. They're a third base pair wobble in effect. So we thought maybe if we run an assembler that works in amino acid space, all of those polymorphisms that don't affect the coding sequence will wash out, and we'll get better assemblies. So we did the thing that I'm sure all of the people in this room do, like we Googled like amino acid assembly, and I'd been collecting references over the decade since, before this, like, hey, I should probably try amino acid assembly at some point. We tried running 3 or 4 of them. They all failed because, bioinformatics. And then I lucked across a recent paper by Martin Steiniger and Johannes Söding that was miracle of miracles Conda installable. So it was in Bioconda. And if you haven't heard of Bioconda, ignore everything else I've said in this talk and just go use Bioconda. But because that's the single most important thing I have to say, which is that Bioconda, when it works really well for installing software. When it doesn't work, well, bioinformatics. Okay, so we stumbled across this paper, and the software installed and the software ran, which was already a good start. And essentially what the software does is it uses an overlap layout consensus approach to do assembly in protein space. And it's sort of a mixed K-mer overlap OLC approach, but basically it translates all of the reads into open reading frames, and then it does K-mer overlaps and sort of works through the layout and the consensus step. And I actually don't think that this is the important part of the assembler. The other important part of this assembler is, and I'll double down on this in a second, is that it doesn't filter anything at all. So basically, if you put a read in, odds are pretty good it's going to give you an amino acid translation of it. A lot of the time that'll be wrong, but some of the time it'll be some reasonable amount of the time it'll be correct. And that's different from the approach that nucleotide assemblers use, where nucleotide assemblers, when they give you a config, by and large, they want to be really sure that that config they give you is correct. So I would say nucleotide assemblers tend to err on the side of specificity, high specificity, and this amino acid assembler tends to err on the side of high sensitivity and low specificity. So we ran it, and we got sort of absurdly high numbers. These are apples to oranges comparison. This was done with K-mers. This was done with Paladin mapping of reads to amino acid sequences. But I sort of just want to show you, like, roughly, 95% plus of the reads made it into the amino acid assembler. That is utter nonsense, just to be clear. Generally, bacteria, if we're gonna paint with a broad E. coli-centric brush, generally E. coli bacteria have, only 80 to 90% coding sequences, so you wouldn't actually expect mapping rates this high, but we'll take it, because the alternative was to throw away most of our data. Okay. So, and we still haven't really figured out how to represent these data properly. So thoughts on how to represent that would be welcome. Okay, so I think that we wouldn't trust the amino acid assembly directly, but we did some filtering. So for example, we ran checkm, which is a thing that looks for highly conserved core essential genes in bins, and estimates the completeness of genome bins based on sort of matches to these hidden Markov models. And what we found was that the summary of this is basically that the amino acid assembler generally improves over the nucleotide assembler, and that generally the neighborhood-based approach improves over the binning approach. So it's sort of a complicated graph to describe, but basically on the x-axis, each of these dots represents one of these genome bins. On the x-axis is the completeness of that original bin using CheckM based on sort of percent of core genes that are there. And you can see some of the bins that were produced in this paper were sort of below 50% completeness. And then on the y-axis, the blue dots are the megahit assemblies of the neighborhoods of these bins using our spacecraft cats approach. And in general, in all cases, the neighborhood included the original bin. That's a good check, right? Everything was above the diagonal line. Sometimes the neighborhoods didn't improve too much. When we extracted the neighborhoods of the genomes and then assembled them, sort of got the same results. In a number of cases, the megahit assemblies improved quite a bit. The megahit assemblies improved a little bit. And then when we ran PLAS, the amino acid assembler, instead of megahit, and estimated the completeness there, we would see in some cases reasonably significant, sort of 10 to 20% improvement in the bins. So basically what we're doing here to get the green ones is we're going in with these genomes into the metagenome assembly graph. We're extracting the neighborhood of reads, we're reassembling those with PLAS, and then we're seeing that we get bins that look more complete. There's a reasonably long discussion in the paper about this, but basically when you get an incomplete bin, there's a couple different reasons why you might get an incomplete bin. One is the data is not in the metagenome to begin with because it didn't make it through the sequencing reaction. The second is that the assembly didn't work or something like that. And so we think that in some cases the assembly didn't work. That's what's going on here. In some cases the binning didn't work. That's much harder to get at because it's very hard to know how to expand bins. In some ways, that's sort of what our neighborhood query is doing. And then, so the data's not there, it didn't assemble, or it didn't bin. And we don't actually know what the problem is. To the extent that we can identify a problem, it's the difference that we can pin the problem down to assembly. It's the difference between the size of the green and the red bars. In some cases, I would say that PLAS did less well than megahit. That's the red ones, where the completeness went down. And that's just because PLAS is young software. And actually, we did this great thing where I emailed the authors and I said, hey, thanks for your tool. Here are results. Can you help explain them? They said, oh, we found 3 bugs. We fixed it. Can you rerun it? And so we're sort of doing the iteration thing where we're working with them to figure out why their software returns poorer results than nucleotide assemblers sometime. I would also say that our read data set is old. It's from like 2014 sequencing, and PLAS has been designed for more modern data sets. So there's probably some stuff there. Okay. This was pretty encouraging, right? Like in some cases we get dramatically better, I would say dramatically better bins out of this neighborhood approach. So then things started to get weird, right? Now, I don't know how many of you work with metagenomes regularly, but the fun thing about metagenomes is you should never look too hard at them. Because if you look too hard at them, you'll always find a bunch of stuff you don't understand. So we started to look at these assemblies, and in particular, we focused on the GYRA gene. There's no particular reason we chose this gene other than that it's not a member of a large gene family. And it's a DNA gyrase that should be present in essentially every genome. I think is a fair statement. So it's one of the ones that CheckM uses for assessing completeness, and it's one of the ones that the Banfield group used to assess their initial sequencing data. And what we found is that, okay, so when you looked at the genomes that the Banfield group had produced, some of them didn't have any GYRAs, some of them had exactly 1, which is what you'd expect, and some of the bins had 2. And this would generally be considered evidence of contamination or weird binning. Then when we looked at things with GYRA from the PLAS, we found that, yep, some of the bins still had 0. That's hopefully due to incorrect binning. Our approach is still very dependent on the bins that were produced by the original group for the querying. In all of the green cases, we increased the number of GYRAs, sometimes from 0 to 2, which is a little bit weird, sometimes from 2 to 3, which is also a little bit weird. In one case that turns out to be because of low coverage, we actually decreased the number of GYRAs. And I should say here that we did hard trimming of all of this data. we knew that PLAS was very sensitive. So before we fed the reads into PLAS, we did a hard trim at a k-mer abundance of 5. So basically, every piece of data we show from PLAS has at least 5 reads backing it up, which means it's pretty solid from a sequencing data analysis perspective. We would welcome more clever ways to ensure that our data is not total garbage, but that we thought that being really stringent would be a good approach. And we showed that this trend holds true across many genes. So one thing, and I'm just going to put this bug in the ears of people that think about metagenomes, is like, well, we'll come back to it too, but why would there be 2 or 3 GYRAs in a genome bin? Why is that expected or acceptable? Okay, so the story so far, right? So I think assembly might break on strain variation. We found that in a mock community. And then we see that the assembly graph neighborhoods contain additional genes that aren't assembled by a nucleotide assembler, right? So far so good. And then we started digging into what these genes looked like. And here we use this wonderful Bandage software to graph the compact to brain graph that the reads that essentially assembled with PLAS into the GYRA gene. So this is the underlying graph of the nucleotide reads that get assembled by PLAS into GYRA, and they're hard trimmed. So we demand that every base here be supported by at least 5 k-mers or more. And what we see is 2 big things. One is that there are 2 different but closely related GYRA sequences. That's what these sort of parallel blue and green lines here mean. This is probably strain variation. And we see that there's a lot of for lack of a better word, spurs in the assembly graph that probably come from additional GYRA sequences that we didn't pull out due to coverage variation. They're probably low abundance strain variants. And when you go ahead and you do sort of an ordination plot of all of the different GYRA matches, using a hidden Markov model using Hammer to search for them, you find the 2 known ones, the 2 that appear in the bin, and then a big cloud of sort of strain varying amino acid sequence, of sort of closely related amino acid sequences. And we think that these represent strain variants that are sort of proximal in sequence space. We see the same thing with cute sequence structure when we look at any of these neighborhoods. So we have a couple pictures in the paper of this. So basically, even with hard-trimmed reads, many minor sequence variants of GYRA exist in the data set. Okay, so, and then we were like, okay, well, now we have these genome bins. We think it's real. When we look at the data pretty closely with a hidden Markov model, we see that it looks highly conserved. It matches really well. Did we find anything new? When we improve these bins, do we find new function? And somewhat reassuringly, the answer was no. You might be like, why are you reassured that you didn't discover new biology? And the answer is, if we discovered new biology, we'd be worried because we'd have to explain it. Whereas if we don't discover new biology, we say, well, many of the bins were already pretty complete. So you'd expect, in general, the way metabolic annotation works, that most of the pathways would have been there. So maybe we're improving the pathway completeness by 5 or 10%, but we're not really discovering radical new content. The purple is the new stuff that we discover, and then the new KEGG functions that we discover, and the gray is just what was already there. So this is probably why we're not going to get it into a high impact factor journal, because we didn't come out and say, look at all this new stuff we discovered. We actually just showed that our software kind of works as expected. That turns out not to be a path to Nature or Cell, but I'm okay with that. So basically, okay. So this is sort of the end of the whatever biology was in my talk. So we see significant amounts of strain variation that's almost certainly real. We have strong reasons to believe that assemblers are systematically underperforming on real metagenomes, and people should give us lots of grant money so that we can do more research. Right? That's how essentially all of these research projects end, is we need more funding. Okay. We do have some other intriguing hints, and I'm just going to put these out. I'm happy to talk more about how we did this if you're familiar with the Sour Mash software. That's what we used. But we've been looking at some of the large data sets like the Tara Ocean metagenome data, and we discovered what I would regard as disturbing hints that strain variation, that assembly is underperforming quite significantly on many of these large data sets. Here, for example, we looked at all of the k-mers in the, we looked at all of the k-mers in either 10 or more Tara Ocean samples or 2 or more Tara Ocean samples, so between 7 and 50 billion k-mers, and we asked how many of them were in the reference databases and how many of them were in the assembled and extracted genome bins that have been published by several people. And basically, we find that even when you assemble from a particular data set, you're getting in the neighborhood of only 80%, between 80 and 90% of what's in the data set. Sorry, 80 and 90% of what's in the data set doesn't come out the other end of the binning software. We think that probably one reasonable explanation for this is that the assembler is not doing well in complex ocean metagenomes. The other thing, and these are hard graphs to explain, so I'm happy to talk through them with a whiteboard or something later. But we asked, like, how, when you look at these data sets, these Tara Ocean data sets have well over 150 samples. There's the bacterial fraction ones that we're looking at. And you look at sort of how much of the sample is known as a function of how cosmopolitan a particular k-mer is. That is, if a k-mer is present in 200 data sets, does that tend to be in a reference database or not, or assembled or not, versus 150 versus 100 versus 50. What we see is that the more frequently a k-mer shows up across all these samples, the more likely it has made it into an assembly. Right? And you can think of that as sort of the obvious statement, like if you wander around the ocean in a sailboat and you dip a bucket into the ocean, if something shows up in all of the buckets, it's likely to make it into a reference genome. Whereas if you only see stuff in the Red Sea, maybe it's unlikely to have been sampled before. Okay, so this is the sort of expected graph. And then the slightly unexpected graph is that when you normalize for the abundance of these k-mers, you see that by and large, most of these k-mers are not cosmopolitan. Most of these k-mers only show up in 5 to 10 samples. So essentially, our argument from this graph would be that this really nice fat line, once you remove the normalization to 100%, out here you're getting something like 0.001% of words from the Tara Ocean data set are in any reference genome or any assembled genome. So the question then becomes, what's all of this stuff? And I don't know. When we look at it, it seems like it's probably minor strain variants of well-known ocean microbes, which just suggests to me that two things. One is, continue with my theme of this talk, strain variants aren't assembled that well, but the ocean's pretty well mixed, which is sort of what we'd expect from the way these samples were taken. And so there's a lot of diversity there that's not making it out the other end of our bioinformatic pipelines, but it's probably not going to be impossible to get at a lot of it. Again, hard to explain, but I'm happy to talk you through. So basically the two takeaways from these graphs: content that's present in many samples is more frequently known, but the vast majority of content is unknown. Again, I don't think any microbial ecologist would necessarily disagree with these statements, but it's nice to get some hard data from some of our approaches. Okay, so now it's time for the conclusions. So a couple of conclusions. So first of all, I think microbial ecologists would generally expect there to be substantial strain variation in any microbial community, in any complex microbial community. There's no one genome in the wild. In fact, this is rarely explicitly stated unless you go talk to people who do a lot of culturing, and they know they're removing true community variability through the process of culturing, and that's accepted, and it's just sort of understood. On the converse, and this became really clear at the JGI user meeting a couple of years ago, when you publish an assembler, what you do is you talk about how well the assembler reconstructs a single contig. And in particular, sort of the sequence yield. What's the NG50? What's the length, and so on and so forth. And I think, sort of in line with what's going on in the human genetics world, this is probably the wrong question at this point. We really want to start looking at population structure in a more hard-edged, hard-nosed way. So having an assembler that throws away 80% of your data in order to make a more confident, specific claim about one consensus sequence is a choice. We should probably make that choice more explicitly. That's all I'm really saying. And I think that this is becoming clear as we move into looking at viruses and metagenomes, which are much more highly variable even than bacteria. We're looking at long read versus short read data. Mick, I don't know if he'll talk about this, but Mick has a really nice paper out where they sequenced the same rumen sample with long read nanopore and short read Illumina. And they showed that there was actually pretty good correspondence there. I'm really interested in digging into his data and asking, of the genomes that came out of the nanopore, how much sort of population structure is missing from those simplified versions. And then, as we get to deeper and deeper sequencing, we're going to see more and more minor sequence variants present in the data. That actually has biological value in terms of looking at selection pressures in the wild. It would be of real value to learn how to extract that and work with that. Okay. So here's the thing that I was saying to some people before the talk started. I'm not actually sure any of what I'm doing matters. And the reason for this is that I actually don't know if the strain variation is necessarily biologically important. For the results I showed you, we don't have any indication that it is. We just see that microbes exist in complex mixtures. We knew that. I guess my main argument as a bioinformatician is that it's probably bad to discard high confidence strain variation unknowingly. It's kind of a bad idea to have tools that just throw it away. And I would argue that the right place to keep it is actually at the assembler level rather than at the mapping level, because you can't recover a lot of polymorphism via mapping. And I would certainly say that it's hard to detect and hard to understand. This has been 3 years of software development and theory development. And I think we have a lot more work to do. So I'm really enthusiastic about our software. I really enjoy software. I think that calculating assembly graph neighborhoods quickly and efficiently could be really useful. Some of the human genetics folk were kind of interested in efficient ways to represent, dominating sets on human genome size things for comparative genomics and so on. The direction we're heading in a software level is doing things like validating bins, annotating bin genomes with local population structure, and then looking at local phylogenomic structure. So there's a lot of work that's going on in this area with k-mers, and we're hoping to bring this into more of a graph perspective. We've run it. So if you work on metagenomes and RNA-seq, one of the fun things is that RNA-seq data sets are like super cute and simple because like coding sequences just aren't that big compared to like expressed gene sequences just aren't that big comparatively. So we've been doing things like building compact de Bruijn graphs for RNA-seq reads, and then using spacecraft cats to sort of collapse them down into simplified structures. We haven't yet found a use for this. We suspect there will probably be something to do with isoforms and queries and complex data sets, but happy to chat. The software is available, and we are manpower, women power limited in my lab. And then something that I'm really enthusiastic about that didn't make it into our first paper, but that we're hoping to write up soon is you can actually build hierarchies of these dominating sets. So you can build a single node, a single dominating node to rule them all, break the assembly graph down into hierarchical components, and start to look at large-scale structure of assembly graphs in a way that I haven't seen done before. So what use this will be, I can't exactly tell you, but this is sort of the technology wing development of my lab, which is like, let's build a bunch of software and then run it on stuff and see if it works and does anything interesting. We call this a cat list because it's spacecraft cats. And then since they set me up to give the first scientific talk of this meeting, I thought I'd weigh in with one or two sort of higher order thoughts. So the first is there are large gaps between what biologists think tools are doing and what the tools are actually doing. I don't think this comes as a surprise to anybody in this room, but it is a real problem. And I think that as the volume of data involved in all of these analyses goes up and up and up, I struggle to analyze it. I struggle to even do the first round of analysis, much less anything more interesting. And I think that we're so reliant on the tools that we have that the biologists are becoming increasingly lost. So it's one of the reasons I put a lot of effort into training. But I think we should be thinking about that when it comes to publications as well. There's this really obnoxious quote, all models are wrong, but some are useful, that's attributed to George Box. It talks about statistical models. It's well known in physics. I really worry about the fiction that we call metagenome assembled genomes, right? We go into a metagenome, we pull out some genome bins, they look good, we reach metabolic conclusions from them. Are they real in any sense of the word? I don't know. They're certainly useful, but I don't know. And I think the vast majority of our inferences about metagenomes come from this kind of analysis. So there's a bunch of amazing papers, especially from the Banfield lab. I have no reason specifically to doubt them, but we also have no good way to independently validate or corroborate them. And so I worry about that at a sort of field level, not at a individual publication level. And they're making their way into genome databases, so that's a problem. So there's this great set of quotes from our otherwise not so great Secretary of Defense from a couple administrations back. Rumsfeld, the known knowns, known unknowns, and unknown unknowns. And I really like looking at the known unknowns and the unknown unknowns. And I just want to say that what's known and unknown depends very much on your background. And in highly interdisciplinary sciences like bioinformatics, you might be talking to somebody and simply not realize that they're coming from a completely different, training background than you. Yeah, I'm going to finish on time. You don't have to worry about it. Last slide, I think. I would really like to see a lot more power tools for digging into large data sets comprehensively without strong priors. So mapping independent and assembly independent approaches. And that's where a lot of our recent work on things like Kevlar, Sour Mash, and spacecraft cats is heading. So I'm having fun, whether it means anything I don't know, but that's sort of the life of a bioinformatician anyway, so I'm good with that. And on that note, thank you very much for listening. I'll just preempt. I'm happy to take one or two questions while the next speaker comes up and gets ready. Uh-oh. I have an enthusiastic hand raiser. That generally means trouble.
I'm more of a biologist. Could you tell me how different are the two gyrations that you showed? How big is the difference in terms of amino acids or insertions?
So at a nucleotide level, almost everything I showed you is within 2 to 4% amino acid average nucleotide identity difference. So the graphs were of the nucleotide identity, not the amino acid identity. At the amino acid identity, it's usually in the 1% range.
Because gyrus is a spot for selection in simple processing.
Yeah.
Some bugs resist to some antibiotics by mutating gyrus.
I did not know that, but it makes a lot of sense. Yeah. So we should look at that.
Yeah, give the list of the other genes that makes the same polymorphism.
Yeah.
So I have one on the protein assembler. How does it handle translation? Does it translate short reads in all frames? Does it use the same translation table? What about when you use it have a lot of Archaea? How's it not?
Yeah. At the moment, I don't believe it uses alternative codons. And I believe what it does is it translates in all 6 frames, and then it looks for overlap layout approaches that yield a longer open reading frame. So it does pick one, but that's it.
Thanks for the great talk. So you mentioned about doing co-assemblies. So okay, I'm not sure if this is a very generic question. So do you think doing a single sample assembly would probably reduce the complexity of strategy variation?
So thank you for the question. I given the amount of time I took to go through my slides, I'm glad I didn't put this in, but now I'm sorry I didn't put it in as a supplementary slide. So we had many suspicions because we were finding that the TG lab was finding that co-assembly was giving much worse assemblies on soil. So basically exactly what you're saying. And you can make a very simple statement, right? The De Bruijn graph never gets less complicated as you add more samples, right? And so we see exactly this, and we've been trying to figure out how to quantify it, and we haven't done that. One of the directions we hope to head in with spacecraft cats is actually to sort of complicated, but essentially combine all the data, and then take analyses of the single data sets, go in and look at the combined De Bruijn graph and pull out sort of, if I had to coin a new term, sort of pan-metagenomics, pan-genomic metagenomics or something, where you're looking at the neighborhood and how it changes in different samples. But one of the motivations for that is that clearly combining samples is worse than sequencing individual samples deeply, analyzing them individually, and then trying to do pan-genomics on the result. It's exactly what you asked, yeah.
Before we start, last question.
Okay, sorry. Anyway, I'll be here through tomorrow evening. I'd love to chat with people.
So, I'm always wondering about the difference between strains in nature and actual just mutations. So, because it could be stable strains or it could be just something that is happening at that precise moment in time. So, I'm wondering whether you've done this data set maybe twice in time to see if it's the same, similar, completely different, or even maybe the best thing to look at this would be to take one of these mock communities that you actually grow in the lab and do it several times.
Yeah.
Growing it several times.
Yes. So mostly we've looked at spatial variation and not temporal variation or replicates. There's a lot of literature in the evolutionary microbiology. I used to be colleagues with Rich Lenski, for example, and we would talk about stuff like this. And I just don't know. But yes, that's certainly a direction we want to go in. I don't have any results yet.
Yeah, I'm coming from the same field as Lenski, so yeah.
Thank you.
Okay, thank you, Titus. And again,